With the remarkable progress in cancer research, we now know a lot about cancer than ever before. But still, cancer remains the deadliest and most feared disease of all. Cancer is so complex that it remains a nightmare even for the best doctor in the field. 33-year-old mother leading a very healthy life, presenting with a bowel obstruction. Her diagnosis revealed that she had a metastatic colorectal cancer. Cancer has already spread to her liver. She decided to fight this disease with everything she had and survive this ordeal for her 1-month-old baby. Her doctors decided to remove her primary tumor from her colon as it was intact. Then they treated a metastatic disease in her liver with doublet chemotherapy. She experienced a high level of toxicity, and with the presence of RAS mutation, a genetic defect in her tumor limited her further therapeutic choices. They discontinued her current chemotherapy. Her prognosis was very poor, and her doctor estimated her survival to be under a year. After careful consideration, her doctors decided to treat her with a liver-directed therapy called selective internal radiation therapy. Lo and behold, her cancer disappeared from her liver. Every follow-up images showed that there was no cancer in her body. She was so happy that she was going to live her life fullest with a child and family. Unfortunately, after a few months, she developed a metastatic disease in her ovary, which was then surgically removed. Later, she developed a lung metastasis, which was also successfully treated with radiation, followed by a surgical resection. Subsequently, she developed a brain metastasis, which was also again surgically resected. Just as she was thinking that she had fought through this and finally succeeded, the disease came back once again, and this time it had spread all over her body, and she died shortly afterwards. Have you ever wondered why it is so hard to treat cancer? In this video, I'm going to give you the overview of how complex cancer is and how we can tackle it, and what are some of the recent advancements in the field. We all assume that cancer is a single entity that is growing uncontrollably. Well, not quite. Under the microscope, cancer cells may all look the same. But the trouble is, even a single tumor has multiple clones of genetically different cells. This is called tumor heterogeneity. There is a heterogeneity within the same tumor in a patient, and there is a heterogeneity between patients as well. In other words, cancer is not a single entity. They are constantly evolving and constantly changing. This makes cancer a moving target. It's like every time we are fighting a different enemy. The fact that tumor is constantly changing their genetic makeup is one of the reasons why it is hard to treat or cure cancer. A treatment targeting one tumor mutation will not work in another. That is why they say cancer is not a single disease. It's a collection of different types of genetically altered mutant cells. Each respond differently to different therapy. If every tumor cell is the same, we would have cured it long back, but it is not the case. So what causes this diversity? Cancer is a disease of DNA. It starts with a series of mutations in certain genes in our cells caused by carcinogen or random errors in our DNA, which leads to uncontrolled cell division. Once a cell turns into cancer, the first thing gets broken is its DNA repairing system, which leads to genomic instability. This turns cancer cells into a genetic rebel. That's when part of our body turns against us. These cells turn into an extreme survival machines and cannibalize other cells for their survival. With the broken DNA repair system, along with the fast cell division, cancer cells accumulate more and more genetic errors in their DNA and produce more genetically diverse clones. Also, cancer cells hijack some proteins in our cell which are responsible for our genetic diversity and use them to change their genetic code to diversify and evolve further. So why do cancer cells need more genetic diversity? Well, more diversity means more adaptation, and more adaptation means more survival skills they can have. More diversity they have, better they are at dividing faster than its neighbor and gaining nutrition, and more successful they are as a cancer. million years of evolutionary history behind this adaptation. Each clones, even in the same tumor, have different survival advantages. For example, some are resistant to treatment, and some have the ability to escape immune attack, and some have the ability to invade and colonize other organs, forming metastatic disease. Remember, metastasis is what mainly killing the cancer patients. So over the time, a tumor accumulates many mutations and branches out into clones and subclones, more like a tree with many branches where each new branch is a result of a new mutation. More the branches, poor the prognosis or treatment outcome. Unfortunately, today our current medical practice completely ignores this information about tumor heterogeneity while taking clinical decisions. Currently, cancer treatment is based on the information acquired by a single tissue biopsy done at the time of diagnosis. But the trouble is, the tissue biopsy that we take is just a poor sampling method that captures just few clones of the tumor, not all. It is like just blindly taking few color beads from the bowl containing mixture of all colors and assuming they are all the same. Similarly, information from a single biopsy does not represent the complete genomic landscape of whole tumor in a patient, and any treatment based on that initial biopsy might not target all clones in a tumor. Because as we discussed, each clone responds differently to different therapy. And also, as cancer is constantly changing, the information from the initial biopsy will be outdated after a few months or even weeks down the line. Due to these limitations of current biopsy method, any therapy based on initial single biopsy might just cut the branches of the tumor tree, but not the trunk. This might give an impression of a good response initially, but the truth is, these treatments are just killing only sensitive cells and leaving out resistant clones, which will then repopulate, and that's when we get recurrence. When they come back very aggressively and spread to other organs. Therefore, targeting a single clone or single mutation will not be enough in a highly heterogeneous tumor. As Dr. Siddhartha Mukherjee described in his famous book, The Emperors of All Maladies, it is like blowing hard on a matchstick long after it had ignited a forest fire. Usually, our immune system is very effective in recognizing and killing any abnormal cells. For example, if any cell is infected with a virus and expressing an abnormal protein on its surface, it will be killed. Similarly, each mutation in a tumor is recognized by our immune system. But unfortunately, they are put to sleep by cancer cells by various mechanisms. And also, cancer cells are wearing an invisible cloak to evade immune attack, sort of flying under the radar of immune surveillance. Very clever. Although we have immunotherapies that can enhance immune recognition of cancer cells, they are sometimes not enough. And there are many ways that a tumor could escape immune attack. Recently, scientists found rare immune cells present in the tumor that are capable of recognizing and killing all types of tumor clones in a patient. Scientists believe that harnessing the power of these immune cells would be the holy grail of cancer treatment and overcome tumor heterogeneity. Dr. Charles Swanton and team are trying to isolate these unique immune cells and multiply them and boost them in the lab and give them back to the patient so that they can target all the mutations and clones in the patient. But no matter how effective these immunotherapies are, sometimes they would still be not enough on its own to tackle constantly changing and evolving cancer cells. Therefore, we should also find and target evolutionary vulnerabilities present within every single tumor clones and stop them from evolving further and progressing. To do that, we need to capture complete tumor heterogeneity profile of an individual and reconstruct the clonal lineage of the tumor. Basically, find out the order in which the mutations occurred, which are the truncal mutation and which are the branches, etc. In other words, we need to reconstruct the complete tumor clonal tree, if you will. But it is not that easy to capture the genetic information from all the clones. As you can imagine, it is not possible to biopsy every single clones of a tumor. So how do we do that? Is it even possible to do that? Turns out it is. There is a new and emerging technology called liquid biopsy. As the name suggests, it's just a quick blood test by which we can now capture genetic information from every single clone present in a patient. So how does it work? Our blood contains DNA and RNA from normal cells as well as from the cancer cells. DNA from tumor cells regularly shed into bloodstream, and using liquid biopsy we can capture genetic information from all clones of a tumor. With the help of next generation DNA sequencing and intelligent computer algorithm, we can now document cell by cell genetic differences and mutations in a tumor and reconstruct clonal tree. Once the tumor clonal tree is constructed, we can then find and target the common genetic vulnerability or mutation present in every tumor clones. Instead of just cutting the branches, this approach will root out all cancer clones completely from the body without leaving any residual disease. Using this method, we can also understand which clones have high metastatic potential and which clones have risk of developing drug resistance, so that we can target those high-risk clones first and stop them from progressing and spreading to other organs. We can also predict the next evolutionary move of cancer cell and change our treatment accordingly. Another advantage of using liquid biopsy is that we can monitor response to a therapy in real time. If we find new mutation in blood, we can switch treatment or plan imaging in advance. This allows us to be one step ahead of cancer. Remember, catching up with cancer is very difficult and usually end up in treatment failure and progression. Do you recall the protein that we talked about earlier, which cancer cells hijack to create diversity? Well, researchers are trying to block these proteins. This will also help prevent clonal diversification and further evolution of cancer, and therefore stopping drug resistance and progression. And these are some of the revolutionary approaches in the pipeline to treat or even cure cancer. So what do you think the future of cancer treatment would be like? For sure, we won't have one magic bullet to cure cancer, but we might use a cocktail of evolution-based targeted therapies and personalized immunotherapies specific to each individual to target and eliminate every single tumor clones from the body. If these researches proven to be effective in the lab and clinical trials, one day we might have a potential cure to conquer this immortal illness and save lives.